very exciting, and I'm really happy to do that with you guys today. And beyond kind of, the amazing scientific tool that gene editing is, which we'll get into in a minute, it's also very much an ethical, situation, given the ability to profoundly impact our society and humanity. So, I want to walk you guys through the science, but I also want you to be thinking about, where this can take us in the future, and kind of, where do we draw the line here? Next slide, Tom, please. Thank you. Okay, so, what is our kind of CRISPR gene editing journey going to look like today? I first want to do a little bit of a science recap and just give an overview of some key concepts that will help with understanding CRISPR. And then I'll define and explain, I think, a little bit more kind of, what even is gene editing, which will bring us to the exciting part of CRISPR as a whole, and going through some of the specifics there and the specific functionalities of how it works and operates. And then we'll take a look at some of the practical applications of this technology, and finish up, as Tom mentioned, with some Q&A and broader, ethical discussions. So next slide. Okay, so there are two really fundamentally important topics to cover here, and the first is DNA, which many of you probably remember. It stands for deoxyribose nucleic acid, and it's essentially just the blueprint of each one of us. But more specifically, it's the genetic material within each one of our cells. And as you can see from the double-stranded helix here, it creates this ladder-like structure. And that ladder is made up of 4 different base pairs or nucleotides, which you can see here are the yellow and pink, and then the 2 pairs, the 2 blue pairs. And Tom, if you just hit a click, but not the next slide. Yeah, there we go. Perfect. And then there's a set of those base pairs is what will make up a genetic sequence or a gene, right? And that is what encodes for something like, blue eyes, or your blood type, or worse, like particular diseases. And that gene is what essentially dictates a particular characteristic in our bodies. And how that works more specifically, though, is that the gene then produces a protein. And with the combination of other chemicals and proteins, that then forms the tissues that will actually make those eyes blue or brown or whatever color. And so the body is comprised of, thousands and thousands of genes. And so they're incredibly integral to what we're talking about today. And it's also important to note that in humans and in other, mammals and organisms, this DNA remains in what's called the nucleus of the cell. And so think of this as kind of the core or the lockbox of the cell. And in these types of organisms, it cannot leave that nucleus. So next slide. And the second key concept that we have to talk about today is RNA, which is ribonucleic acid, which structurally, as you can see here, just looks like half of that DNA ladder. And it's also comprised of 4 base pairs. They're the same base pairs or nucleotides as DNA, with the exception of 1. But RNA is actually what translates the DNA sequence or that gene into its actual function. So it's incredibly important in the role of protein synthesis and of gene expression. And there are many, many different types of RNA. And one that you've obviously likely heard of recently, of course, is mRNA. And this is the type of genetic material that is used in the Moderna and the Pfizer COVID vaccines. And mRNA is important here today too. And mRNA stands for messenger RNA. And its sole purpose is to basically, carry a message based off of the DNA sequence. And this is because, like I mentioned, in humans or in other eukaryotes, your DNA cannot leave the nucleus. And that's what makes it so safe and so strong. And so the mRNA, what it does is it takes the information from the DNA, messages it out of the nucleus and into other areas of the cell so that it can be read and turned into a protein. And that protein then gives rise to a function of that gene, right? We talked about like blue eyes or blood type. So that's just a little bit of the science overview. I just wanted to make sure that we kind of talked a little bit about DNA and RNA because it is going to continue. It is going to apply to what we're looking at. So next slide. Okay, so this is a really important distinction, I think, to make, is this difference between genomics and genetics. And so let's first take a look at genetics here. And so it refers to a particular sequence of DNA, right? So that one piece, not all of it, but just one piece that encodes for a molecule. And it specifically looks at a single gene, right? That one sequence of DNA. And that might be causing a disease in this case. And that disease might be caused by a hereditary gene or something that was passed down by one of your parents. It also could have been caused by a mutation that occurs naturally, right? So we hear of mutations and we kind of freak out. We think of, some large mutant beast. Mutations happen every single day in all of us. Sometimes they mean nothing. Sometimes they do, and they can lead to diseases. And so then on the other hand, we have genomics. And so genomics is much, much more broad. And so it's all of the genetic information within an organism, all of the genes, all of the DNA, right? So if we use us as an example, it would be all of my genetic material, all of my little pieces of DNA together. So it really just takes into account everything. And it's the study, when you think about somebody who studies genomics, it's the study of genetic information of an organism or, again, as a whole versus a particular disease or sequence. So the important takeaway here is that a gene is just a stretch of DNA. It's just a piece of it, and it codes for something in our body. And that's incredibly important when you're working on CRISPR because you're trying to change that. So think of a gene as kind of just like a little mini recipe. And so what happens when a gene is mutated, you can either be born with this genetic disease, or I said it could happen, through unknown significance. That's exactly where gene editing comes in and can make this correction. Okay, perfect. Thanks for going to the next slide. So let's think about gene editing as a whole, right? What is it? And I'll be honest, in straightforward terms, it's just as it sounds. It's literally the ability to edit a gene, whether it's one or whether it's many. And it's an incredibly powerful tool that allows for the manipulation of a genetic sequence. So it will ultimately alter the expression or what that piece of DNA will become. So it's pretty important. And I've always liked to describe it as the ability to just copy and paste DNA, right? Cut and paste. And so what you're doing is you're just changing the code of DNA to change the outcome, be it a disease, be it a trait. Really, you can kind of do whatever. And that's where some of the ethics comes in. And there are three main tools that are used for gene editing today. CRISPR is probably the one that everybody has heard of the most. It's kind of the most popular. It's kind of pop science nowadays, and rightfully so. It's a fascinating thing. And then there are megatels, which is a slightly different approach. It's what I have focused most of my research on, have been megatels. And then the last one, and I would say for sure the least common, is what's called a zinc finger. But the last two, so megatels and zinc fingers, I'm not going to go in today. I think we're just going to focus on CRISPR and how it works and how its structure is and where it'll take us for the future. So again, gene editing, it just involves changing that DNA sequence, thereby changing its message. So you can go to the next slide now. Perfect. Okay. So we've arrived to the point of what is CRISPR. And I'm sure many of you are wondering, what does this stand for? Because it is an acronym. It's a bit of a tongue twister, so bear with me. It stands for clustered, regularly interspaced, short palindromic repeats. You don't need to know the specifics of it. It's very jargon heavy. But what it is from a high level is it's just a powerful technology that alters and changes, again, a particular sequence within a gene in a very precise way. And this means that it can correct and fix a mutation in a gene that gives rise to a disease, like we've talked about. And when you think about it, I mean, it's kind of a pretty powerful technology when you think about how many genes are in the body, how many things could go wrong. To be able to have a tool that can precisely target and change something is pretty revolutionary. So we're going to have to go back into a little bit of science. So bear with me. And so I'm just going to talk a little bit about how CRISPR actually works from a high level. And so it works by a protein that's called Cas9. And that Cas9 protein, which you can see here on the slide, is that light blue amoeba kind of looking like shape. It first will find the sequence of DNA that we call the target sequence, right? What it's trying to find. And then it literally will go in, once it finds it, it'll cut the sequence like a pair of scissors. And it knows exactly where to cut by being programmed to recognize a particular sequence. You could tell it to target any sequence you wanted. It's totally up to you. And so when you program that sequence, you're using a strand of guide RNA. And that is what matches and will recognize the target DNA sequence. So think of it like a puzzle, right? You're putting in a strand of RNA that's only going to stick and that's only going to match to a piece of DNA. And so once it finds that target sequence through a chemical reaction, it causes a cut in the DNA at that desired location, right where the gene is properly, not functioning, right? And from there, you've got a few options. You can either leave it as a plain cut or a splice acceptor, as the two ends are called, or you can insert a new piece of genetically created genomic material, so a new piece of DNA, however long, however short. And this enables the cell to literally function properly again. And so this can result, like I mentioned, in a gene being fixed, like a disease, where you can stop the disease from expressing, or it can cause a gene to have a different function. And so that's, again, it's a pretty powerful technology. So you can head to the next slide. Okay, so here what I wanted to highlight is sort of where the link comes in how does this work with humans and what's the actual application. So after that high level kind of how does it work, let's take this to the application. And there are two approaches, and one is called ex vivo, which is here on the left, and that stands, that kind of means out of the body. And the other is an in vivo, and in vivo means like within. And so in the ex vivo example on the dark blue side, this works by extracting cells from a patient, right? You're going to take the blood and then you modify these cells in the lab using your CRISPR technology that we just talked about. You introduce that into the pool of cells, and then you grow and expand those cells from that patient so that there are more than just a few, right? In these instances, you need a lot of cells, in the millions, if not billions. And then the final step in the ex vivo process is to reinfuse it back into that patient's body through a process that's called transfusion. And what happens is that these reintroduced cells will engraft into the body, or they'll kind of be, absorbed and accepted by the body. And all of the subsequent cells will divide and will contain that edit, that new edit, and will no longer have the incorrect or broken sequence of that gene, which is insane. Like, it's amazing. And so my previous company uses this exact method, and it's currently in clinical trials with patients. And it's how our product Zynteglo that was taken to market a year and a half ago, it's exactly how it works. And so, that drug is basically working by modifying the autologous hematopoietic stem cells. And so that means just the same person's blood cells to treat sickle cell and to treat transfusion-dependent beta thalassemia. And beta thalassemia is a really horrible, severe anemia or iron deficiency disease. And sickle cells, as some of you may have heard, is a disease. It's a genetic disorder, and it turns certain of the red blood cells into these sickle or moon-shaped cells that when they travel through the bloodstream, they can get stuck and they can kind of all pile up. And because of that, it creates some of the most horrific pain that humans can really experience. Shortened lifespan. It's a horrible disease as well. But the interesting part of sickle cell is that it was essentially it came out of evolution, because if you have sickle cell, your body cannot get malaria. You are immune to malaria. And so at any rate, you can probably imagine that personalized medicine like this is incredibly expensive. And it is. Our Zynteglo drug product that works on beta thalassemia is $1.8 million a patient. So it's not a cheap therapy at this point whatsoever. So then on the right, we have the in vivo. And the in vivo is a different process. And it's exactly what my particular area of research focused on. So I'm a little biased. I think it's cooler, but it's really not. It's not too dissimilar. And so how this works is that you use a vector. And you can think of a vector as sort of kind of in this case, like a capsule. And this can be in the form of a nanoparticle or a virus. And so when you use a nanoparticle, which is what I used, it's just a small molecule that's made up of a bunch of lipids or a bunch of fats that essentially replicates a cell. So it's a tiny little small molecule. Or on the other hand, you could use a viral particle, which is kind of crazy to think about because you are literally using a virus. Now it's a completely denatured virus, but you're not going to get sick. It's not how it works. But these little particles and these little viruses have the amazing ability to infiltrate the body. And so they are such a great way to get editing into the cell, because that's one of the hardest things, is to make sure that you're getting the edit into the cell. The body is incredibly smart. Anything that comes in, it sees it as a foreign object and it will fight it. And so you have to use different means of tricking the body in order for it to work. So once you've sort of selected either of these two vector types in the in vivo process, either the lipid nanoparticle or the viral or virus-based vector, you're able to deliver, again, that CRISPR-Cas9 to the targeted cells or tissues within the body. Again, via infusion in the example, like the ex vivo example, as a direct injection. So that route of administration, whether it's a transfusion or an injection, is very just specifically based off of the disease you're looking at. I'm not able, unfortunately, to go into too many specifics of my particular research just because the projects are still ongoing. But I do want to highlight two other really cool studies that are still in trial that use this to sort of help explain how CRISPR is working today. And so the first one is from a company called Editas, and they're using, again, the virus-based method or the adeno-associated virus that will carry their CRISPR-Cas9 technology. And they will be directly injecting, believe it or not, into the eye for a therapy targeting a disease called LCA, which is a rare disorder that impacts the retina and causes like severe acute visual impairment. And so that's a pretty powerful solution to have for patients with a rare disease that doesn't have any other solutions. And then the second company that's also doing another, I mean, there are many companies right now doing gene therapies, but another one to highlight for sure is a company called Intellia. And they're working on a therapeutic for FTA, which is an inherited, condition of a buildup of a particular protein in the organs and the tissues. And they're using the other option. They're using the lipid nanoparticle that encapsulates their CRISPR technology. And what happens for them is this will cause a knockout, as it's called, which is essentially just a removal of the production of the disease-causing protein in the liver. And so what happens there is the body then creates cells that no longer produce the protein that's building up in the person's organs. And so, that's a huge therapeutic advancement for a very rare disease that has no other option. And so both of these really, I think, to me, highlight the ability for patients' lives to be enhanced and to be bettered because of these rare, debilitating, and degenerative diseases. And I do want to mention that there are still challenges that need to be overcome because delivering CRISPR-Cas9 to a sufficient number of target cells to actually get that therapeutic impact is a major challenge. You need to have enough edited cells that go back into the body in order for it to work. And that's a huge challenge for many, and myself included. And you can't just throw more edited cells into the body because you run the risk of having an adverse immune reaction that typically in these cases is lethal. And another big issue is the possibility of what's called off-target edits to the genome. And this is where CRISPR goes and it goes rogue, right? And it just starts cutting everywhere. And so all of a sudden you have splices in all different parts of the genome, and that causes a lot of issues as well. And so there's still clearly some advancements that need to be done here, but a lot of progress, as we can see, has already been made and is still out there really helping a lot of patients. So next slide. So this is where a bit of the ethics comes in. And there are two different ways in which gene editing can be done, and both of them have very different outcomes. And so the first is the somatic approach, which is where a somatic cell, which is just, think about it like a general cell, right? It's your skin cell, it's your organs, it's anything that hasn't differentiated, like a neuron or a blood cell, which is a very particular type of differentiated cell. And somatic gene editing only affects the patient being treated. This is really important. It only changes their cells. It doesn't change the subsequent offspring or lineages. So for example, if I were to get a CRISPR gene therapy, it would only impact me. It wouldn't impact my offspring. And so somatic gene therapies involve modifying a patient's DNA to treat or even, cure a disease caused by a genetic mutation, just like the one we saw, on the last slide, where you again extract the tissue or the blood and create an edit, then reintroduce it back into the patient. And this is the one that's far more common. And then we have the germline editing, which is a bit more blurry and where the ethics kind of comes in. And this is the process that uses a human embryo to alter the genome. So remember, the genome is all of the DNA, not just one specific gene or one specific disease, right? It's everything. So you can choose to edit whatever you want, but it will affect everything in there. And so the result is that human that's born has a genome that has been impacted as a whole. And This editing is done at the very earliest stages of embryo development, which is what makes it so successful down the road. And so, like I mentioned, this germline editing will affect everything, every cell in the organism. It'll include the egg, the sperm, the blood, the brain, everything. And it's passed on to future generations. That's the major distinction. And this is a huge impact, not only to the patient, but to their offspring and to the offspring's descendants. And so this edit will just continue to be passed through the generations, essentially indefinitely, unless, say, like specific mutations occur throughout someone's life. And that's really, I think, where the major ethical considerations do come in. So again, so the somatic cell gene therapy has been very well studied over the last few decades. It's really the one that has the powerful therapeutic capabilities. And there are regulations surrounding it. There are a lot of regulations when it comes to scientific research. But germline, on the other hand, is relatively new in comparison and doesn't yet have those regulations at this point. So that's certainly a consideration to think about when we talk of ethics surrounding CRISPR and gene editing. Next slide. And so just quickly to highlight again the benefits and the disadvantages of this, I'm going to present a real life case of germline editing, but I do want to highlight some of the good and bad first. So this is obviously, like we've seen, it's an incredibly beneficial technology that gives us the ability to change the future of diseases. And that's such a fundamental success in the field. But I don't want to use the word cure because it's a very avoided word in the science community, given how definite it is. But this technology very well, prevents, reverses, and even in some cases actually cures diseases. And so from the medical perspective, this is huge. This is a huge benefit. This is a huge advancement. And even aside from those therapeutic considerations, CRISPR also has the ability to be used to develop things such as, biofuels or agricultural adjustments to ensure, crops that are able to grow in dry climates, climate change, et cetera. So that's, again, another important distinction of it. And then on the other hand, there's actually some scientists that are working on editing the genome, again, all of the genes of a mosquito so that they're unable to infect humans with malaria. So there's some clear advantages here, again, along with some ethical, even in the good, but there's some pretty strong negatives. So we'll take a look at that. Next slide, Tom. Thank you. And so I think one of the biggest ones that, where the questions of ethics surrounds is this notion of designer babies, or even frankly, designer pets, that have the exact traits that we want them to have: hair color, facial features, body types, height, weight, like you name it, we can create it essentially with this technology. And we can also ensure that the genetic offspring are, quote, strong, right, for whatever, criteria you're looking for. So that's a major conversation topic. And the science community feels quite strongly that that's not what this is developed for. And then we also have the ability to design virulent diseases. And this is, like a truly crazy mutant virus, right? Think of something worse than what we're dealing with right now. And so we're able to edit and create some pretty nasty things out of this. And so if it's in the hands of those that don't, that want it to go there, that's really where another big concern comes in. Where do we draw the line? What's okay and what isn't? And another issue that this technology brings up, which I mentioned in the slide before, is the germline editing problem, where it will have infinite downstream implications, right? Where all of the offspring will carry that edited person's trait. And so it's almost infinite when you think about it. So that's another consideration to it as well when we look at the germline part of gene editing. And so the last thing that I'm going to bring to you guys today before we open it to questions is a real life case of CRISPR. So Tom, you can hit the next slide. So CRISPR babies, this did happen, and it was a big deal in the science community. Back in 2018, a scientist in China named Dr. He decided to edit the embryos from an in vitro couple, which is a type of genetic, reproductive technology for those who can't conceive. And so that resulted, and he did it to have the babies result in a genetic, a gene that couldn't contract HIV. And so what he did, it was very controversial. He used the germline CRISPR, right? So he used the embryo. And this was such an uproar. And there were many factors of why this was the case, but he kept pretty much the entire procedure quiet until the birth of these CRISPR twin girls. I think their names are Luna and Nana now. So they're kind of infamous twins. They're 2 years old. And so doctors in China, in the US, like all over the world, felt that this was an incredibly unethical, obviously secretive, right, and poorly conducted use of this technology that's still not ready to be used, frankly, in even a lot of experiments from a somatic standpoint. And so the thought of using this in a human, in a germline procedure, is like the technology isn't even there yet. But it also begs the question of, do we ever want it to be there? And I think one of the major problems that a lot of scientists were feeling is that, do we also know enough about the human genome to be able to edit cells within us and know that the functionality will remain and not cause serious downstream long-term effects? And we know a lot about the human genome. We do. And we learn more each day. Technologies, AI, everything. It's helping us every minute. But we're not there yet. And it's a big question. The human genome is complex. And so to be able to edit one gene and to think that it won't have downstream effects, we don't know enough. We just don't. And so a lot of scientists really felt that this was a huge violation. And not at all what this was intended to be used for. And so this doesn't mean that we won't get to that point where we could, but it's just a matter of when, right? And so I think what we all need to be thinking about when we approach that point is, where do we draw the line between ethics and medical therapies? And how far are we really willing to take this? Is this a benefit or a threat to humanity? And so I'm going to leave you with that final thought and open it up to any questions that you may have on what I've presented.
Yeah, thank you so much, Abigail. Thank you so much.
You're welcome.
So, guys, you can jump in, and if you have questions. And Tom, you want to add something?
No, actually, let's hear from Adrian, please.
Thank you for the presentation. Really insightful. I love the passion as well. My question is regarding so the COVID-19 vaccine using mRNA, and if I remember correctly, I read something in the news that it used AI. Can you maybe elaborate how AI helped discover a vaccine, sorry?
Yeah, so AI technologies, it's not something I'm incredibly up to speed with in terms of the applications of it with gene editing, but essentially, from my understanding, how it's used is they're using it to find the targeting sequences. And so with science, so much of it is trial and error. It's we think this is the sequence, let's try it. Doesn't work, you don't get a hit, try it again. You keep going back to the drawing board. And so what AI is able to do, again, like we've talked about in some of our classes, is it's a needle in a haystack. And so you can quickly identify what that target sequence is of the genome within that virus. So a virus, let's use COVID, right? SARS-CoV-2, the virus has one particular strand of RNA. So it has that half DNA, right, in it, encapsulated in it. And so what AI could be used in this context, from my understanding, is to understand and identify specifically, what that is faster. They also, there's plenty of other sequencing tools that are used. There's, GWAS, which is also another way of doing it. So I hope that kind of helps. I'm not entirely sure how the AI is different from the traditional sequencing of RNA. But one thing that it does do is it just speeds up the process for the therapy to know exactly what that target sequence needs to be without having to go through 100 different trial and errors.
Great question. Are there any other technical questions? Sartak, please.
Yeah, thanks, Tom. Thanks, Abigail, for your presentation. Yeah. It was really interesting. I have a question on just the any side effects of this CRISPR process that scientists are not aware of. And I think you touched upon that there might be some long-term consequences that we don't know about yet, and also a question about genetic determinism and how there's an attempt to cure psychiatric diseases as well, neuropsychiatric diseases, mental illnesses, and how CRISPR technology, whether it's promising for those kinds of diseases as well. So those are my two questions. And also if you can touch upon maybe like government policies, like how they have an impact on this research, because there's a lot of like tension between like science versus religion.
Yeah.
The churches being involved, and questions as like taking over God's role in the universe. So if you can touch on that.
Sure. So I'll start with your first question in terms of the adverse side effects. Yeah, there are absolutely. And, I've seen it with therapies at my own company. It happens with any of these products because, like I said, the body is so incredibly smart. And what it does when a foreign object comes in is it fights it. And there's the immune system, which in my opinion, I think is one of the most complex, difficult, intertwined, just crazy parts of science that has so many complexities. There's so many players involved, signaling pathways. It's nuts. I don't go there. But what happens with gene editing, or what can happen, is the body can reject it. And essentially what happens is it can become toxic. And so the body can't process it if it's too high of a dose. And so a lot of studies have to look at efficacy and toxicity ranges to be able to say, this is a safe range. This range here is approaching a threshold, and everything above the threshold is dangerous or lethal. And so one of the major adverse effects that I would see was what's called a cytokine storm. And it's actually very similar to what happens in a COVID patient. And a cytokine storm is essentially your body's immune system going insane. And so your body has your immune system has all of these different types of cells, your T cells, your B cells, monocytes, a million of them, right? And so what happens is once this foreign object comes in, it fights it. And what can happen is it can overreact to it, and it releases what's called a cytokine, which is essentially like a molecule. And too much of that then shuts down the organs. And so what you see is exactly the same thing with certain very, very acute COVID patients too, is this what's called a cytokine storm. Again, it's the immune system that's just shutting off. And you really can't, unfortunately, from everything that I've seen, you cannot stop a cytokine storm. It's pure toxicity. And so that does happen in some of the the testing before it goes to clinical trials. So again, this is another ethical consideration. Any of these studies do have to go through animal models before they are deemed safe to be put into a human. They have to go initially through usually mice and then non-human primate or monkey. And so there's, again, major ethical considerations. And so one of the other things that we've seen frequently, and this is public knowledge, so I can speak of it. My company, its product is being used to literally, I don't want to again use the word cure, but work on sickle cell. And recently had a handful of patients in that trial get cancer. And so, this is a very new technology. And I think for a lot of these patients, it's a last resort and it's a life-saving thing. And so the adverse effects are certainly part of it. But again, it can really, those adverse effects can really be dependent upon the patient, dependent upon their entire genome, what their composition is. That's why somebody with COVID is either asymptomatic or dies. So that's kind of how the side effects occur. They're there. And science works very hard to, have studies that diminish and ensure the least side effects possible, but they do arise. Your second question was about complex psychological diseases, right? So a perfect example would be schizophrenia. Schizophrenia is a complex genetic disease, and it is made up of, as is autism as well, it's made up of thousands or hundreds of genes. And those diseases are called complex because it's not a particular gene, like blue eyes, for example, where it's called a Mendelian trait, where a gene codes for blank, right? A gene codes for something. A complex neurological disorder contains so many different genes, it's difficult to target what's what. And so that makes it incredibly difficult to target gene therapy or CRISPR technologies at this point for something that takes into account so many different genes in order to give rise to a disease. But the technology may get there. I don't know.
All right. If there are no more technical questions, let's move the conversation into more of an open discussion about some of the ethical issues is suggested by these kinds of technologies. One of the larger issues is about access. And in fact, we already deal with this in a very significant way in the world today, to the point at which there are many experts who consider poverty itself to be a disease. And if you look at statistics based on economic demographics, by every medical measure, it is life-shortening and life-threatening to be poor. These technologies that Abigail has been discussing and that are emerging now are extraordinarily expensive, but they also have the capacity to save lives and to improve quality of life for people who would otherwise suffer from debilitating and in many cases, lethal diseases. So how do we balance these economic concerns, whether in a privatized health system like we have in the US, or in a public health system where the cost of these extraordinarily expensive, sometimes not 100% effective methodologies are borne by the public at large? What are some of your thoughts, please?
I would say with the diseases, and it's something that should be equally addressed. So there needs to be a kind of rebalancing. And when we're seeing something right now with COVID, which is affecting, it doesn't discriminate between borders. It affects rich. It doesn't viruses don't discriminate based on on income. And so it's important for the global community to come together, because if the virus, something like a virus, is in one part of the world, it's going to affect it's going to affect the rest of the world. So I think even when it comes to diseases, it's important for the global community to work together, because if there's a solution that's discovered in one part of the world that could help, it could be, what I'm saying is, it could be in country self-interest to help other countries.
Yeah, absolutely. And one of the problems is too, again, it's because it's so personalized, it's difficult to mass produce this at this point. And so again, the technology, I think, also could be working towards advancing that to make it more accessible. I mean, 1.8 million was considered a reasonable approach to pricing our product. It's not reasonable. So that's, again, yeah, accessibility is a huge thing. And, who gets ahead, right? Who's able to, which races and which countries and which people end up benefiting the most? Who gets ahead? Is this just promoting privilege? That's a big question. It's a big question.
I can jump in. For me, we have to treat the CRISPR like a nuclear. So the people that we need, we can go back. It's like we need rules to play this, and especially, for example, in Europe or in the United States. But in China, they have other rules, and you go to the United States, different rules, Europe, different rules. So how we can make something together, because it's threat to the generation, the human race. It's a threat of human race. Maybe it will be an extension if we use this CRISPR for the mass. I think in the future will be cheaper. So if you use this, you will lose the human race. So we need rules. We need rules like we did for nuclear. We did the same. We have to do the same for CRISPR.
Yeah, absolutely. And like I mentioned, the germline is certainly an area where there needs to be much more regulations. I think, Sartak, actually, you mentioned the question about what regulations exist. And there have been different regulations throughout time. I remember in the Bush administration, there was a huge issue with stem cell research. And it's, again, I was young at the time, but there were some bans on those types of studies. And I believe they were lifted to, I think, in 2015. Don't quote me on that. And so there's there's more regulations in terms of how it's used from a laboratory practice standpoint, but the germline is still uncharted territory. And by and large, the science community seems to sort of agree, in the US and in China, and where obviously this CRISPR baby happened. And in other parts of the world, I think because science, people really want to maintain the same level so that it's comparable, because, there's obviously a lot of issues with certain countries thinking other countries' data isn't worthwhile to trust or to, replicate or whatever. And so that's again, another important thing to keep in mind, is that I would say by and large, the science community does tend to align on regulations so that the quality of data can be comparable. Doesn't mean it always is followed, but I think from my experience, that's what I noticed.
It's also worth noting that all of the biases that we see in other areas of society occur within the scientific community. And particularly problematic today is the Western bias in science. Which greatly limits, and in fact, language itself can be a problem in science, because language tends to silo information within whatever language it was produced in. And so you look at the distribution of scientific information across the world, and for example, there is a wealth of very good data out of China that is simply not accessible because it hasn't been translated into Western languages yet. And you also encounter biases. So there are many scientists that I've spoken to over the years who are quite borderline racist about whose data they trust and whose methodologies they trust.
Absolutely. Yeah. And that was, I think, a big consideration as well in the case of these CRISPR babies. With a lot of the Chinese scientists who were saying, you have tarnished our already, tough reputation with the Western world in terms of our science. And so there was a lot of outrage from that because of that inherent bias that's very real. I experienced it a lot. And a lot of times people will say, well, we're going to have to replicate this data. We don't trust it. And it's like, there is maybe sometimes a very valid reason to replicate data, but don't do it because you don't trust it. Do it because it's important for you to have a better result.
Can I ask the questions? I was a bit late though, but I do have some maybe silly questions. Like people still have rumor about the virus, COVID-19 virus. Is it naturally or is it from the lab? I think that is too mystery to people like me.
Yeah, no, it's a great question, Jan. And they are able to sequence it. And because they're able to sequence it, they're able to understand by and large where it comes from, if it's synthetic or if it's not. You can tell when something is synthetically derived. A lot of DNA is synthetically derived in the lab because it's very easy to do. You can grow it off of different bacteria and whatnot. It's very easy. But this from everything that I have read and looked into and from the science community, it has been sequenced and it has been identified that it was not created in the lab. Again, a huge racial bias, a huge painful racial bias. Yeah, that it did stem from animals, and that is, monkeys, like bats. There's so many different animals that contain diseases, pigs that have things that are comparable to infect humans. And so, that's an important thing to know is like animals carry diseases. Some of them don't affect us. Some of them do, evidently, right?
Yeah, it's good to know. And now I feel good, but also bad. Now I know it's not from the lab, which means not created by human being on purpose as a weapon, right? But now I realized, if I recall to the wet market in China, it's really brutal, and it's just. I hope one day you guys could go to China, definitely visit those wet markets to understand the culture, and I hope those wild animals could disappear eventually in the market.
Yeah, absolutely. No, it's important to not cast judgment on these things, right? This could have happened from any country. And it's important to know that, again, a virus doesn't care. A virus doesn't see country or race or privilege. Its sole purpose is to replicate and to spread. And it doesn't care who you are or what you're doing. Its job is to grow, and it will do everything in its power to do that and to continue to mutate to do that as well.
Which I think is a really profound point for understanding the biological landscape overall, because that's what all living things do.
Yep.
And in fact, it is entirely reasonable to look at higher order organisms, animals, ourselves, as essentially very sophisticated machines with no other purpose except to spread our genes. It's a very interesting perspective for looking at the world. I'll also add that it gets even weirder than that, because not only do organisms compete to spread their genes, genes actually compete with each other under certain circumstances within an individual living organism. It is a very strange world out there.
Yeah, absolutely.
It's such a fascinating world.
I know. And honestly, when you think about science and you think about genes and you think about, you know, having been in science and working on all of these different diseases, you genuinely wonder, how are any of us normal? That is just a constant question of it's pretty profound. So yeah, I mean, the competition of genes is again, it's another issue. You know, it's why things like sickle cell were you know, came about. And so again, if you have sickle cell, you're immune to malaria. So nature basically safeguarded against those who were getting it and created this natural occurring mutation. So Sartak, I see your comment. You know, again, I'm sure there are plenty of people out there, including the ex-director, who believes that. I think until there's truly concrete, viable proof that it was genetically derived, to me, it's a pretty racial claim.
So if I may, let's pivot for one more philosophical and ethical discussion here. At this point, the technology that we're talking about is essentially exclusively limited to treating diseases of various types. And we can manipulate gender. But it's an inevitability that in our future we will have the capacity to manipulate traits, at least to some degree. So where do we draw the line? Is it justifiable to choose the gender of your baby? Is it justifiable to specify an eye color or a hair color? How about a height? How about intelligence? How about muscle mass? Anything else you can imagine? Where do we draw the line with designer babies? What are your thoughts?
Well, it's an interesting question. We already doing that to the animals, right?
Like, are you saying with in the example of like dogs, Jan, where we have like purebred?
Yeah, purebred dogs. Yes, that's what I'm saying. And also like masculine or more meaty chickens.
Mm-hmm.
You know, things like that. We're not eating. We're not in natural world already. We're
far beyond.
It's a more ethical issue about human beings.
So where do you think the line should be drawn with humans? Or do you have any thoughts, Jan?
I have no idea, to be honest, and I'm pretty sure there's some countries maybe are already doing something. I don't know, but I'm just assuming maybe like how to say that the baby, like if the parents cannot give birth to baby by themselves, they need to do how to call it in English more.
It's called IVF, in vitro fertilization. Yeah.
Yes, but how do you call that? I mean, like 20 years ago is something very new, and I remember in China at least when that baby was born, it's such a big news.
Mm-hmm.
But now it's so common, and parents who ever cannot give birth to babies naturally, they could just use this methodology to have a baby. Yeah. Like 20 or 30 years ago, it's so unethical, but now it's just so common in our life. So I think life is just being invaded eventually, step by step.
Yeah, just to answer the question of Tom, I think, we will never go back. And it's like the continuity of the it's not the progress, but it's like in the future with climate change, all these things, we will have like politicians, they will justify to change the human race, because we will not be able to adapt especially for the climate change, and especially we are talking about Mars, we are talking about space. So the human today is not adapted for the space. So this technology with time will be like minimum things to do, like vaccine. If you want to travel, you need vaccine. You are agree or not, you are not agree. It will be the same. If you want to travel, for example, in the space or with climate change, all this changing are coming. So we will change ourselves to adapt. And the objective, as Abigail said, like a virus, we want to grow. It's the growth. If you survive, you will do everything to survive. So if someone tell me, if you change your genome, you will survive, I will do it. So how I think it's like the logic, the things for the future. It's very sad, but
I also want to add on that it's important to also consider like human suffering and pain, and if these technologies can relieve suffering and pain, and like Mohsin was saying, extend human life, it's always like a risk reward calculation, and also who has the authority to make these kinds of decisions. I was just reading an article on how people with disabilities, there's an argument that these technologies make them feel like they're inferior, and that their certain conditions should be like removed from the human race. And so there's definitely an ethical kind of component that needs to be discussed.
So that's a really important point, Sartak, and thank you for bringing it up. I would recommend, there's a film that has recently been nominated for a couple of Academy Awards. It's free on Amazon Prime, entitled The Sound of Metal. And the premise of the film is that there's a rock musician, a drummer, who goes deaf. And you get a glimpse into the deaf community. And this issue comes up precisely because one of the places that he goes for support is very clear that we do not view deafness as a disability. And in fact, if you get into what we refer to as the disabled communities, you will find that phenomenon very frequently, where people reject the idea that it's a disability or a disadvantage, and they reject any approach to their problems that try to re-normalize them. And so, for example, one of the, I shouldn't give away any plot elements in the film, but for example, in the deaf community, we now can use cochlear implants, which bypass a damaged ear and are able to more or less directly stimulate the brain. And that can give people whom otherwise would never have a chance at hearing anything the ability to hear again fairly intelligibly, actually. That technology is massively controversial in the deaf community. In fact, there are many deaf people who consider those who get that implant to essentially be traitors to deafness. So it's a very, very complicated issue. And that's the question, is if we can manipulate genes, how much variety in human expression, in human phenotype, do we allow? How much uniformity do we require? It's a new level of eugenics, potentially. What do you think?
Yeah, I think it's a great point, and I think it in some ways comes down to the disease, I think, or the disability, right? And deafness is not a disease. But I think, I think back to some of the patients that I've interacted with through my career, very short career. But I remember a group of sickle cell patients, and people who have sickle cell tend to come from sub-Saharan African populations in places where where malaria is most prevalent. And so it's very, very dominant in Black communities all around the world. And so, those patients, one of the girls who was in this trial, a young woman, she said, I'm scared to even trip because it'll create an acute sense of pain throughout my entire body and I'll be hospitalized for weeks. They can't have any impact because of their blood cells. And it'll cause, she said, the pain that I get is something like 10 times that of surgery. And she said one morphine or one, painkiller does nothing. It's like 10 times, like it's just the worst. And so when she received this drug product for sickle cell, there was a 60 Minutes episode on this 2 years ago. And she's now running around, and there's photos of her running, going for a jog, something she never could have done. She was taking up Taekwondo, and you see a scene of her literally being tossed in the air and slamming down on the ground, which could have previously put her in the hospital for, frankly, a year. She now lives a pain-free, essentially sickle-free life. So I think, in her context, it's relief, it's hope, it's not having to be in the hospital for half the year because of her disease. So I think it does come down to the individual and to the condition at hand. But it's an important thing to remember, it's not because there's anything wrong with you, right? It's hopefully there to be seen in society as a way to make your life better.
Yeah, I agree with Abigail. And just to respond to Tom, the problem, if you go to the history, always the people that they want to introduce the new technology, they give you examples like this. Like if your father has Alzheimer, you would like to use this technology. Of course, I will use it. But the problem, for example, I give you an example of MOG. They invent MOG just for the explanation is for hunger. So is to eliminate hunger. But these guys like Monsanto, they use this MOG just for money. And the hunger, we still have hungers. So all the technology in the history, they give you just an example, it's good for humanity, it's good, and you give you an example that people, they are suffering, and now using this technology, they are very happy. But it's just an example. But the objective is to make money. And in the end, they don't resolve these problems. And you have a lot of, it's like Facebook, for example, to connect people, blah, blah, but in the end, just to make money. So you give you an example, special example of that man, they are suffering and they use this product, they will survive. But you have just one example. But in the end, the result is to make money.
And, I as an environmentalist, I appreciate very much that you brought up the gene editing of agricultural products, which has been going on in both natural and laboratory-driven ways for decades now. And what has that resulted in? Not an appreciable change in world hunger. What it has resulted in is absolutely appalling actions by the big agricultural companies, where for example, farmers who grew their own wheat, but a neighbor, sometimes miles away, grew one of these proprietary wheat brands, typically from Monsanto. The pollen blew and pollinated my wheat, and now Monsanto is suing me for theft. That's happened all over the world, right? What are they doing with these genetic engineering technologies? They're doing things like making sure that the crop can't naturally produce seed, so that you are dependent on buying seed from the company every single year. This is actually one of the great untold stories of the invasion of Iraq from 2002, is all of the deals with American agribusiness. The Iraqi climate is quite unique, right? This is literally the cradle of civilization. Okay, the Tigris and the Euphrates rivers right there. And they have, from the beginning of human agricultural technology, been cultivating specific varieties of wheat that are adapted over nearly 8,000 years of selective breeding to fit that climate perfectly. Well, Monsanto had a contract with the US government that had them introducing their proprietary pay-to-plant varieties of wheat, replacing these heirloom varieties across the country. And that's something that gets almost no coverage in the media, right? In fact, a few years ago, Monsanto is in so deep with the US government that a few years ago, the entire state of Nebraska passed a law forbidding journalists to report on these issues or take photographs of farms. That law was since struck down by the US Supreme Court, but it's absolutely crazy. My environmentalist rant notwithstanding, Mohsen brought up a critical point, and maybe this is a good juncture for us to bring this conversation to a close, although there's clearly much, much more to be said. Humans have been innovating technology since we learned how to harvest fire. Agriculture itself is one of our greatest inventions. The pace of technological innovation has been accelerating for the entirety of human history, and it is now at a fever pitch. And we should ask ourselves, what does this change? We can look at history and ask the question, what has this changed? What has genuinely gotten better? What hasn't? What problems still remain? And, since this is a philosophy club, let's bring it back to a philosophical point. What has technology changed about the fundamental human condition, about the ways that we are born, we live life through happiness and sadness, satisfaction and dissatisfaction, never-ending wants and desires, and then inevitably we die? What does technology do to change those fundamental realities? On that note, my friends, thank you all so much for being here and participating in this conversation. In particular, thank you to our guest, Abigail, for stimulating a truly great event. And of course, there's so much more to be said. So hopefully the conversation will continue beyond this meeting. Thank you all so much. Great seeing you.
Thanks, guys.
Thank you so much.
Thank you.
Thanks, guys.
Bye.